bottom of console's iterative programming this week. I really do.
Oh, iterative.
Yes, it iterates and causes my construction to bounce around, and then gives me, well, off results. I think it's adding them together rather than just telling me the final movement. It's adding all the movements together. So it's a problem. I've been trying to read the manual.
And all else fails, read the directions.
Yeah.
Well, the directions are obscure and in tiny print. So I've taken to putting them in a Word document so I can read them.
Well, Pakhi. All right. Welcome to the meeting. I don't know if Pakhi wanted to give an update on her progress. If she's ready to do that yet.
Hi.
Hi.
Yeah, so I've been working on like generating the graph and stuff. I'm using like the code from the NDP repository, and like I've edited some functions like the train file and NDP file, but like I'm facing a lot of errors right now, so I'm trying to fix that. So nothing much to update. Like I've just been fixing errors for the past the weekend. So
All right.
I will let you guys know once I have some results.
All right. So yeah, you're going to work on that this coming week then.
Yes.
Are there any other things that you have on your timeline that you need to address, or?
Nothing as of now. According to the timeline, I would be like I will work on the graph neural networks only, and like I've been writing some content for the blogs as well, which I'm planning to post in the coming weeks.
Okay.
Yeah.
Well, that sounds good. All right. That's great. Thanks for the update. Now, I don't know where Mehul is. I know he's been busy the last couple weeks, but hopefully we'll get an update from him as well. We got an update from him like 2 weeks ago, but it's always good to keep up with, keep going on the project because we don't have a lot of time this summer to get things accomplished. So, so I guess we'll start off with a data set that I found this past week and is just published recently. And what they're doing in this paper and data set is they're trying to characterize the L1 worm. And so the L1 worm is right after hatch. And so this is when the egg hatches and it unfurls and it becomes this very small version of the adult. And, then, of course, the worm goes through several larval stages, L1, L2, L3, L4.
Which worm are we talking about?
This is C. elegans, although some of the other nematodes do similar things. They have similar larval stages. So that's, and then L1 is the site of some sort of plastic response. So there are a set of experiments you can do where you can immediately just right before hatch, you can put the eggs in like 15 Celsius buffer, which is like a stable liquid, and then put them out on a plate, and then they'll hatch, and they'll stay in this arrest state. So they won't eat any food. They'll undergo starvation, but they can live like that for a while. And so this has consequences later in life. You can extend the life of the worms, and they have this plastic response to starvation. And then there are some other plastic responses later on in larval development. But this is focusing on L1. This is just, I think, just normal L1 C. elegans biology that we're going to be talking about. So let's see. So this is the paper here, a full body transcription factor expression atlas with completely resolved cell identities in C. elegans. So the significance of this paper is that it adds to the data set that we have for the embryo. So we went, in the last couple of meetings, we went through the embryo, what we have in terms of data, which is this three-dimensional positional data on each cell, and they're these developmental cells. So we, have a developmental lineage. And then at the end of last meeting, sort of after the meeting, if you go to the recording, I went over the interactive C. elegans lineage tree. That's something that can be found online. And that will allow one to map from those developmental cell lineages to the adult cells. Okay, now I'd like to talk a little bit about L1 arrest, or L1, and the properties of an L1 worm. So we talked about L1 in the main meeting as being the first larval stage of C. elegans. And actually, this applies to all Caenorhabditis species. I know C. briggsii has an L1, and so this is a picture of the adult worm, and it has eggs, and most of them are hermaphrodites, so they fertilize their own eggs. They lay a bunch of eggs, and when they're fertilized and they start developing, you get this early embryo, which is a cluster of cells. The later embryo, you start to get the shape of a worm. It's coiled up in the egg, and then the egg hatches, and you get this L1 phenotype immediately upon hatch. And then, of course, you get your stages L2, L3, and L4. And of course, L1 is much smaller than L3 or L4. And so each one of these stages, of course, we get this post-embryonic morphogenesis. We add cells, so L1 starts with like 663 cells, and you move up to the 959 in the adult. And this is for the hermaphrodite. So you gain a lot of those cells in L3 and L4, hence the difference in size. Actually, the size is due to growth. There's some cells along the seam that are actually growing. So this is we're being added to the worm. So, but L1, of course, has some interesting properties. As I mentioned in the main meeting, you have this phenomena called L1 arrest. So this is where you hatch the eggs in an environment with no food, and you can arrest this phenotype. You can arrest the L1 phenotype for a long while. You can starve the eggs in a medium and a buffer, and you can put them on a plate, and they will survive without food. And they go through this starvation phenotype, and it's a form of plasticity. And so they can persist in L1, but then it has effects down the line later in different larval stages and then in the adult. There's also this dower stage that begins at L2 and goes to L4, where they grow a very thick cuticle, and they're able to protect themselves from adverse environmental conditions. The same with L1 arrest, that's due to adverse environmental conditions. So you have a lot of plasticity in the larval worm that you don't necessarily see in the adult, and you don't necessarily see in the embryo. As we mentioned in the main meeting, the lineage tree is invariant. In other words, unless they're genetic mutations or we're dealing with genetic mutants, the pathway to different cell types is basically deterministic. But you do have this organism level plasticity that's very interesting in C. elegans development in the larval stages. So this brings us to this paper by L. Ryan Baugh, and he's done a lot of work on looking at L1 arrest. This is to grow or not to grow. Nutritional control of development during C. elegans L1 arrest. And he's done these experiments where they look at L1 arrest. This is a response to starvation conditions when eggs are hatched in an adverse environment where there's no food or where there's limited food. And this is a form of diapause. So in invertebrates and in insects, we have different forms of diapause. And so this is a part of life history where growth is arrested, and it has effects in later life, but there's a trade-off between survival and growth. And so he says here in the abstract, C. elegans larvae can also be reversibly arrested. He says in the abstract, C. elegans larvae can also reversibly arrest development earlier, during the first larval stage L1 in response to starvation. L1 arrest, also known as L1 diapause, occurs without morphological modification, but is accompanied by increased stress resistance. Caloric restriction and periodic fasting can extend adult lifespan. So this is a form of caloric restriction, and as we know in humans, there are mixed effects with respect to that. But this is something that we want to maybe study in C. elegans to see what the effects are for humans. So we can study nutritional control of development using the L1 arrest model. We can also look at aging and diseases like diabetes, obesity, and cancer. And of course, these are all sort of NIH-centric interests. So these are things that we want to know. We want to use a system with well-worked out genetics, and so that's why we're doing this, making this parallel between C. elegans and humans. And so he talks about, this paper kind of goes over a lot of the work that's been done on L1 arrest, and what goes on at the molecular level, and then what goes on in the phenotypic level. So the nervous system, epidermis, and intestine contribute systematically to regulation of arrest. But cell autonomous signaling likely contributes to regulation in the germline. And so there are different effects in both the germline and in in metabolism, in cellular growth. And so we know, for example, that there are a small number of genes affecting starvation survival. Many of these genes are involved in L1 arrest, but they also affect adult lifespan. So then we have this paper, which is the L1 stage. So we have the data for the embryo, we have positions, we have the lineage, and then we have for this, we have cell identities in the adult form. So, at L1 stage, you have all of the cells that differentiated during embryogenesis are present, and there are going to be more cells that are born that are post embryonic, but that's not really important for this. What this allows you to do is map L1 to the embryo with the positions of the cells, and then also it offers gene expression information. So I'll show you some sample data and maybe what you can get out of those data later. So this is in Nature Communications. This is, of course, in C. elegans. And so the abstract reads, invariant cell lineage in C. elegans enable spatiotemporal resolution of transcriptional regulatory mechanisms. So basically, because you have these cell lineages that always unfold in the same way, we can then look to the resulting adult cells and their position in the embryo, and we can do single cell gene sequencing or labeling, and we can get information about gene expression both in the embryo and then in the larval stage in the adult. So that's where the power of kind of putting together these C. elegans data sets comes from. So this paper talks about a tool, and they're developing this tool called RapCat, which is robust point matching and piecewise affine-based cell annotation. So they use that to automate cell identity assignment. So what they do is they have these microscope, the image stacks. They take the L1 larvae, they put it under a microscope. They take these images, and then they're doing this point matching and piecewise affine-based cell annotation. So they look at the position of the cell, and they can identify it, and they put a label on it. And so that's what they're doing here. Now, in the embryogenetic data set, we have what we call cell tracking, and we do this here too. And the cell tracking is basically where you take microscopy images and you take the movies, and there's a software that allows you to sort of reconstruct the paths of the cells, and it tracks the cells and it can identify them. And so that's a feature of a lot of these full embryogenetic full larval data sets. So that's that's convenient for us because it gives us data that we can actually use and build images of, and I'll show you what those look like later.
So do they have the sizes of the cells?
Well,
This.
we'll see in a minute. As somewhat. Well, yeah. So, but what they're getting at here is they want to identify the cell, position of the cell relative to other cells, and then the expression values. So they're first, they're not like segmenting cell boundaries in this kind of work. They're just getting it down and dirty, sort of information about the positions of the cells and what's going on in them. How can you label them? So yeah, they're not that interested in getting a really refined size measurement or shape measurement for that matter, which would be nice because I don't think the data set exists. It may be a little bit beyond the technical scope of what kind of tools we have to really get a good map. We could probably get a lousy map. I don't know if that's helpful though.
Are they 100% correct in identifying the cells?
They get pretty correct, but I don't know if they're absolutely. I have to look into the methods, but they do this, on a number of specimens, so they're able to get like pretty decent information. I'm not sure what their accuracy is though, or how they calculate that. So basically, they have these image stacks. They have profile reporter expression. So this is where they have a fluorescent signal for a certain type of certain gene, and then you have an intensity of that fluorescent signal, and that tells you what the relative expression of a certain gene. And what they're looking at here are 620 transcription factors. So transcription factors are just things that get expressed by a gene. You can have multiple transcription factors per gene, but these are important transcription factors, important mRNAs that we know, maybe are well characterized, and so that's maybe the most interesting stuff. Although, there are all sorts of transcription factors, and there's this whole project called ENCODE, where they look at gene expression and function and all that. And some of those data are in C. elegans, so that's why I bring that up. Transcription factor profile-based clustering analysis defines 80 cell types distinct from conventional phenotypic cell types and identifies 3 general phenotypic modalities related to these classifications. So this is not like the labels. You have the sort of the labels that we talked about in the lineage tree, and then it becomes like the adult cell, it becomes like a neuron or a muscle cell, and there's a nomenclature there. What this is they're actually identifying the cell based on transcription factor profiles. So this is where you get the transcription factor activity in each cell, and then you try to classify them, not based on what tissue they're in or their sort of lineage, but what they're expressing in terms of transcription factors. So this gives us a couple of ways to classify cells. And so, they found a couple of patterns or a couple of groups that are, interesting. First, transcription factors are broadly downregulated in quiescent stage hermaphrodite-specific neurons, suggesting stage and cell type specific variation in transcriptome size, which is interesting because that suggests that, during transcription, there's certain depending on the cell you're in, or even like when the cell is active versus quiescent, that there are differences in what gets transcribed. And it's just, something you can identify from a study like this. And, the issue of function is another question. But that's what they found. Second, transcription factor expression is more closely associated with morphology than other phenotypic modalities in different pre- and post-differentiation developmental stages. So this means that transcription factor expression is associated with morphology. So if you have, a certain type of cell with a certain shape, and we know kind of from not characterizing it with sort of in a numeric way, but just knowing the morphology of the cell, because we know the morphologies of the cells. We can describe them in terms of morphology. It's just that we don't have the data set where those things are numerically represented. But anyways, this is something that we can use to sort of classify cells as well. Finally, embryonic cell lineages can be associated with specific transcription factor expression patterns and functions that persist throughout post-embryonic life. This study presents a comprehensive transcription factor atlas for investigating intra-cell type heterogeneity. So this is, they're using single-cell RNA-seq, which is this next-gen method, and they're able to take single cells and they're able to do this RNA-seq. This is the typical method for this. What they're doing here, of course, is this a different type of method using like fluorescence microscopy, basically. And so this basically they're saying here, while cell typing has historically relied on cellular phenotype, which could be, functional, physiological, developmental, single-cell RNA-seq, transcriptome capture with unsupervised or marker-guided clustering to organize cells into biological meaningful groups is revolutionizing our definition and perspective of cell types. So phenotypes of C. elegans cells have been well characterized at single-cell resolution, including the invariant cell lineage throughout development. However, accurate and comprehensive whole-body determination of individual cell identities by transcriptional profile is a challenge that we don't have, we don't have the answer to. So people have done single-cell RNA-seq in C. elegans, and there's some thorough molecular atlases of embryogenesis in the adult nervous system. And these are not, these are kind of, in some ways, inaccessible, because what they tend to give you are these UMAP image or visualizations. And what I'm going to show you in a bit is this sort of table I can extract, which is very different from that kind of an atlas. And we had this conversation on Friday in our open source meeting, and I know Dick was there at the end, but I don't know if he caught it. But it's basically where, a lot of groups will do a lot of data collection, and they'll make the data public. And they'll put it in this atlas that is maybe what you might call a high-level representation. And what that means is that I can query like a UMAP visualization for a certain gene. And I don't necessarily know how to interpret the UMAP visualization. And it doesn't give me numbers. But I can see that it's, in the mix, which is a little bit, I don't know how useful that is. I mean, it depends on what you want to do. If you want to be comforted in the knowledge that you're going to have, like that gene is in that cell, or what how the genes are clustered in a certain cell, then that's great. But if you want to get, if you want to build alternative models, or you want the numbers, then you're out of luck. And a lot of groups will, build these atlases, and then maybe they'll build an API, but probably not. And it's hard to access the data. And we had a back and forth about this Friday, so this is not something that no one thinks about. It's just something that's a unfortunate fact of working with data and making it public. So yeah, and then of course, that aside from that problem, they point out that in these molecular atlases, the number of resolved transcriptomes represents approximately half of the total cells in each investigated system, but are considered robust largely due to the transcriptomic homogeneity among symmetric cell pairs. So they might take like one side of the, usually the cells in C. elegans, the adult cells, are symmetric. You'll have one on the left and one on the right, say. And then, those are sort of sister cells. They may or may not come from the same lineage, but they function sort of on the left and on the right. You see this in the C. elegans connectome a lot with neurons. But, they might take like one of those pairs and sample them and say, well, it serves basically the same function, so let's try it and see. And so this is a problem of sampling. So they're offering kind of a different way of doing this. In this work, we applied a topology preserved matching model to automatically annotate all 558 cells in 3D image stacks of L1 larvae. So there are 558 cells in the L1, or at least you can annotate 558. There are actually, I think, 600 and some, but that's what they're using here. Then use image analysis to profile reporter expression for 2/3 of worm transcription factors. So image analysis is being used instead of the RNA-seq method. And the RNA-seq method, of course, gives you really good resolution in terms of different transcripts. Well, what this does is it ties it more closely to the annotation of cells in situ, which is good. And then you can actually get, you don't maybe get as much specific information about transcription factors, but you can, create a probe that has a specific sequence and get that. So they're able to do this in our transcription factor profiles. We found numerous instances of differential transcription factor expression within cell types defined by cellular phenotypes. And so there's this intratype heterogeneity, which is really interesting. You may or may not think that's interesting, but if you think about like the way we understand cell differentiation, of course, we think, well, certain things need to be upregulated or downregulated to have a cell, be stable in its fate, maybe. But of course, neurons are very diverse. Muscle cells may be less diverse, but what they're saying is that you have different things that are up or down regulated depending on different aspects of that morphology. So depending on function, but also maybe depending on, cell cycle or time of day or whatever. And this is not a great insight, but, having the numbers to back this up is good. And so, so they talk about their results. And, I want to get into I'll get into some of the images here. So this is the worm here. This is a microscopy image. This is, I guess, a side view. This is where they can create the point cloud or the point matching. So they have these point matches from their fluorescence signal and the cell bodies. They can match those. They can update the transformation function. And they build this map between these two sets of images. And they get a pretty good accuracy here. Annotation, okay, here's the question. Vic asked, annotation accuracy, it's within between maybe like 0.9 and 0.999, but then there's some variation here depending on how they're doing this. So, this is the bipartite graph here, and they're just matching the cells, and they're getting this kind of accuracy. So not horrible accuracy, but not perfect accuracy. Okay, so I guess I want to get on with some of the other things I'm interested in with this paper, why I'm interested in it. One reason is because they have data that they offer. And so they have a couple of Zenodo repositories. This is their Cell Explorer version 1. This is their Atlas. So they actually have this in a Docker file, and they have it set up so that you can download it and run it. And this is on Zenodo, so this is like, just kind of an archive. This is RapCat version 1. This is robust point matching and piecewise affine based cell annotation. This is I guess they're running this in MATLAB because they're M files. But this is where, they're running some of these. They have sample data, this bipartite graph that they're running, the RPM, the calculations for that, the transformations, and so forth. So you can do all those things. Okay, then you can transfer from MATLAB to basic data types. And then you have a training set where you have training data and you have testing data and everything you need. So that's great. But one thing you can do, of course, also is you have access to some of the training data and some of the testing data. And I'm going to show some examples of that in a little bit here. So they have a number of data sets where they're basically taking the cells, they're tracking them in the images, and then they have these output files that are training their model. And so what you can do is take those data, you can extract them and turn them into tab delimited data. And once you do that, then you can get it into tabular form, which I have here. So the reason I'm using a spreadsheet for this is because it makes it very intuitive and easy to see what kind of data we have. So what they have is they have basically very similar data to the Watterson data set that we have for C. elegans embryogenesis. We have the name of the cell, and now in the Watterson data set, which is the one I've pointed people to for GSOC, you have the developmental cells, and those are going to be like A, B, D, C, the sublineages, and then the nomenclature. These are different. These are adult cells, and these, at some point, those developmental lineages, basically there's a differentiation step, and then that's it. And you end up with these adult cells. So it's actually somewhat easy to take your developmental lineages and map them to these adult cells. And they're always the same. They always have them fold in the same way, so that map is more or less invariant. There are some exceptions, but that, it's not important right now. You also have your XYZ coordinates, so when they take images from the stack, they'll have like an image in that two-dimensional space of the image focal plane, and then you have a stack is basically a series of focal planes stacked on top of one another. So in the focal plane, you get your XY coordinate, and then in the stack, you get your Z coordinate. So wherever it is, going up or going down. So that gives you a number that you can then have that gives you an XYZ representation of that cell. That's the centroid of the cell, but we don't want to say it's the nucleus, because the nucleus is pretty large compared to what they're characterizing here. And it's where the fluorescent signal is, sort of the center of mass for that. So that may or may not be directly in the center of the nucleus. So we'll call it a centroid in the sense that it's that center of mass or center of gravity for that fluorescent signal. And so that's what we have here. Then we have intensity data. So we have maximum, mean, and standard deviation of intensity. And this is just the fluorescent signal. And then you have this RGN size, which is the region size. This is in voxels. So this can be maybe interpreted as, well, this is basically the fluorescent signal in voxels. So I didn't run the analysis on this. It doesn't look like there's a correlation between mean intensity and our region size. But, I don't know exactly what the correspondence is, but they have this. This is not cell size necessarily, but it's just the region that you have in voxels. So this is potentially interesting number of voxels, potentially interesting information. You have this mass. I don't know what this is. Then there's color. So you have red, green, and blue, and these are the fluorescent signals and the intent, or the sort of the color components of those. So we have a lot of information here. And then this is just one piece of data. So there are other types of data you can get for this. They have other, I didn't put it in here. I just wanted to show an example of what these data look like. Now remember I said this was a larval stage, C. elegans. This is L1. So if we go back to this, I actually put this into a visualization. I put this into Scilab, and I actually prepared some plots where I, graphed out some of these. So this is kind of a messy graph. This is supposed to be like a surface graph, but I didn't get it quite right. But this just kind of shows some of the geometry of the L1. So this is the anterior end, this is the posterior end. And you kind of see how it looks kind of like a worm. This is another angle of that. So, I have to perfect this one. I have to get make a surface and see if I can do that, testing it out. This is a point cloud for the L1 larval worm. And so again, you get this sort of the anterior end here, the posterior end here. And it doesn't have a lot of structure. Kind of almost looks like the embryo, but it'd be interesting to map from the embryo to this L1 stage. This is another perspective on the point cloud. So you can take these point clouds, you can rotate them once you plot them out and see what they look like. They're going to be areas that are more dense with these centroids, these fluorescent centroids, and some that are less dense. And that relates to the geometry of the cells and the distribution of cells and so forth. You expect there to be it to be more heterogeneous in the larval and adult worms, simply because they have tissues. So you have a lot of cells, and you have a lot of nuclei in muscle. You have a lot of nuclei in the connectome and so forth. So you can see that kind of structure already there. And then this is another perspective of the L1 worm. Again, not like high resolution when you have point clouds, but there's more data to be added here. I didn't plot every piece of data available, but this is interesting. So then, you can actually color code these points with different attributes. So if you're interested in gene expression for certain genes, or fluorescent intensity for whatever reason, you can get that information out and plot it out and get a sense spatially of what's going on. Maybe also some spatial statistical, like if using spatial statistics to find out things about certain regions of the worm, like if there are things along the body wall or in the connectome that are interesting, that might also help with that. All right, so it's like we have Alexander. Hello, welcome. And it's like, yeah. So did we have any questions or comments on that?
Yeah, Bradley?
Yeah.
And I'm a little confused. If you know the transcriptome the way they got it,
Yeah.
when you identified the cell.
Well, probably not. I guess that's the question. Yeah. Well, I mean, you can get a pretty decent probability of knowing it, but the thing about the transcriptome, yeah, it's like there's a lot of heterogeneity, and it's like predicting a cell from the transcriptome is probably going to be really hard to do. I know people want to sell it as being easy, but it's not. Like, because you have things are expressed differentially in different cells of the same type. So if you have a neuron, there are all sorts of different expression profiles for neuron, even for the same cell over time. If you're measuring it during the day or in the evening, or when the worm's quiescent versus active, and then of course if you depending what part of cell cycle the cell is in, well, it's not so much of a problem for the differentiated cell, but yeah. And then, so there are a lot of conditions where the transcriptome can vary. It's kind of hard to make that prediction on what specific cells.
Go to the extreme of saying that the transcriptome is irrelevant to the cell type?
No, well, no. Well, we'll talk about another paper where that they're kind of making the case that it's essential to differentiation. But it's just kind of hard, because it's kind of like, if I show you a bunch of beads in a jar and I say, is there a pattern? Identify a pattern in this set of beads. Like you can identify a lot of potential patterns, and maybe none of them are relevant. Maybe one is relevant. There's a lot of conditional expression. So like remember I said that L1, you have this environmental plasticity that happens where the cell undergoes starvation, and where the worm undergoes starvation, and the worm can persist sort of in a rested state for a long time. Well, that involves a lot of gene expression. It induces a lot of gene expression change. So the gene expression of a starved or an arrested L1 worm is going to be far different from a normal L1 worm. And that's, And there are probably gradations within that. So it's not that it's irrelevant, it's just extremely hard to know kind of what the conditions are, and it's very that measurement is very susceptible to any set of environmental conditions that the worm is in.
Okay. Well, I'm reading a book that was published last year called How Life Works.
Oh yeah,
By Baugh, you know the book?
I've heard of it. Yeah.
Oh, okay. I'm reading it, and it's inspiring me to write a book for you, though. But
Oh.
I'll see how it comes.
Yeah.
But at any rate, it seems to me that the book is questioning very much the genocentric view of development.
Yeah.
Okay. And transcriptomes, if anything, are supposedly an expression of the genes. Yeah. So that's why I raise these questions, whether this is going in the wrong direction.
No, I mean it's it's like we have a lot of people have done people have built all these methods for doing gene expression profiling, maybe understanding gene expression, and it's a really hard problem, and it's like we haven't made a lot of progress on it, but that's to be expected. But still, it's like again, I think the thing to stress there is that it's just so highly conditional on the environment, and it's something we're really used to. We talk about genes, especially in C. elegans. We think, well, okay, C. elegans is a pretty easy lift, but it's not necessarily that. Okay. Yeah, it's worth a lot more discussion. But yeah, so Alexander, hello. Sorry I can't speak right now. I'm in a quiet space. I'm new to the OpenWorm project, and so I thought I'd join a meeting. Yeah, welcome. Jesse said, welcome, glad to have you here. Okay, so the next thing I wanted to do another paper, and this paper is on cell cycle and differentiation. So if we go to this paper, we see that what they're proposing is that there's an alternative cell cycle, and that coordinates multiciliated cell differentiation. So this is interesting because they're proposing that there's this type of cell differentiation that's coordinated by the cell cycle, and so they're laying out this idea in this paper. Now, before we get into the paper, I did want to talk about cell cycle a little bit because you're not going to necessarily get where this is going unless we get into it a little bit. So this is cell cycle. You have basically a cell will go through different stages as it divides. It will a single cell with a single genome will divide into two cells. There will be some sort of ring in the middle that constricts the cell. It creates basically two compartments that become their own cells, and then the genome doubles in size, and then those chromosomes segregate out so that at the end of the cell cycle, there will be two cells that have the same complement of things. And some, whatever's in the cytoplasm, sometimes that segregates, sometimes it doesn't. So, for example, you can get maternal factors that go through the cytoplasm. You can get sometimes proteins that go maybe to one daughter cell or another daughter cell. But that cell division is really kind of coordinating all of that. And so we know that cell cycle is also involved in oncogenesis. So for different cancers, cell cycle can be regulated or involved in that process. And so also, with cellular reprogramming, we know that cell cycle is important. So there's this, this whole role of cell cycle, but there are these cell cycle genes, of course, that regulate this process. So you have these different stages. You have G0, G1, S, G2, and M. And so a cell cycle will enter G1, it'll go through S, it'll go to G2, it'll go to M, and then exit in G0 if it's successful. And so, there are these different phases of cell cycle that involve different things. But the molecular network is actually the interesting thing here, because it involves some well-known genes. So, G1, you have these P factors that regulate cyclin D and CDK6 and CDK4. And then as you're entering into S, you get, repression of cyclin E and CDK2. You get this repression in S phase of cyclin A and CDK2. In G2, you get this repression of cyclin A and CDK1. In M, you get the repression of cyclin B and CDK1, and then you exit the cell cycle. So it's basically about building up transcription factors, building up gene expression for these different genes, and then repressing them at the right time so that you can enter the next phase of the cell cycle. Okay, so and then the timing of this is important, because you don't want to spend too much time in G1, or else you have aberrant cell cycle, or too much time in S phase, because you have something that's aberrant, and of course that can lead then to cancer phenotypes. But also, you can have you can modify cell cycle for different cell types. So sometimes, S might be extended, and G2 might be shortened in evolution. And as long as all this is coordinated, you can have different cell types. But yeah, so people have worked out the gene expression pathways here. And so in this paper, they talk about this alternative cell cycle that coordinates multiciliated cell differentiation. This is actually from, I think, Nature. It looks like a Nature paper. But anyways, the abstract reads, the canonical mitotic cell cycle coordinates DNA replication, centriole duplication, and cytokinesis that generate two cells from one. So this is where they're basically, everything gets duplicated, and then you get two cells. Some cells, such as mammalian trophoblast giant cells, use cell cycle variants like the endocycle to bypass mitosis. So what we're describing is mitosis in the last image. We know the cell cycle genes, we know how that works. And it's involved in some forms of, helping along differentiation and cancer phenotypes, which is a form of differentiation, bad form of differentiation. And then we have these cell cycle variants that are maybe things that are not mitotic. So this is endocycle as an example. Differentiating multiciliated cells found in the mammalian airway. So this is in the lung, brain ventricles, and reproductive tract, are postmitotic, but generate hundreds of centrioles. So they don't use mitosis for their cell cycle. They use a different pathway. They're able to divide and produce these centrioles, each of which matures into a basal body and nucleates a motile cilium. So they basically are functional in that way. Several cell cycle regulators have previously been implicated in specific steps of multiciliated cell differentiation. So this is where, we know the cell cycle for like mitosis. We know the genes that are involved in that. We know the timing more or less. And then now what they're saying is that, this multiciliated cell differentiation has a similar type of structure where there are these cell cycle regulators that we can identify. Here we show that differentiating multiciliated cells integrate cell cycle regulators into a new alternative cell cycle, which we refer to as the multiciliation cycle. So this is in a specific cell type. This is an alternative to mitosis, but it operates in a way similar to a regular mitotic cell cycle. And this is, it's an interesting sort of specialized system. The multiciliation cycle redeploys many canonical cell cycle regulators, including cyclin-dependent kinases, CDKs, which we saw in the figure, and they're cognate cyclins. So there are these cyclins and CDKs that get coordinated, and they get coordinated in time. So that's where these things kind of come together, is that they use sort of the same mechanisms, but they use them in a different way. For example, cyclin D1, CDK4, and CDK6 which are regulators of mitotic G1 to S progression. So we saw in the image, we saw G1, that these things get suppressed, or they get like suppressed or shut down, and that allows the cell cycle to move from G1 to S. And if they don't get shut down, then G1 persists and something bad happens or something weird happens. So there's this, you have to regulate the progression to each cell cycle phase. But anyways, these factors here, cyclin D1, CDK4, and CDK6, are required to initiate multiciliated cell differentiation. The multiciliation cycle amplifies some aspects of the canonical cell cycle, such as centriole synthesis, and blocks others, such as DNA replication. So you don't get DNA replication in this alternative cell cycle pathway, but you do get centriole synthesis. E2F7, a transcriptional regulator of canonical S to G2 progression. So this is a different part of cell cycle where you go from S to G2. It's like the next step. Is expressed at high levels during the multiciliation cycle. Okay, so again, there's this parallel between the two types of cell cycle. In the multiciliation cycle, E2F7 directly dampens the expression of genes encoding DNA replication machinery and terminates the S phase like gene expression program. So again, you have to shut down S phase to enter G2 phase. Loss of E2F7 causes aberrant acquisition of DNA synthesis in multiciliated cells and dysregulation, which is, of course, where you have something that's supposed to be maybe like if you want to upregulate a gene, it's not upregulated when it needs to be. And so you have oftentimes people will point to dysregulation as maybe a symptom of disease or a symptom of some disorder or something going wrong in terms of function. That term is used a lot in the medical literature, but it basically means that it's not regulated in the way we expect it to for a specific function. So dysregulation of multiciliation cell cycle progression, which disrupts centriole maturation and ciliogenesis. So again, you get this E2F7, you get a loss of it. And then when you get a loss of that, it affects other things downstream. It dysregulates multiciliation cell cycle progression. That disrupts then centriole maturation and ciliogenesis. So it's this cascading effect. We conclude that multiciliated cells use an alternative cell cycle that orchestrates differentiation instead of controlling proliferation. So this is where in cell cycle, you have this sort of trade-off almost between differentiating and then proliferating. So a cell can proliferate, in terms of the same cell type and just divide and divide and divide, or it can differentiate into a different cell type. And cell cycle is sort of that hinge. And that's what they're saying here, is that in this system, you have that trade-off where you have differentiation or proliferation. Either proliferation is dampened or it's accelerated. And that's, that plays a central role in the whole system. So,
Bradley,
yeah.
Do you have a picture of this?
Well, let's see. We can go through.
A picture of the cells.
Oh. So this is, I don't know what this image is, This is a good one. This is cell cycle phase of basal stem cells. This is a UMAP, so it's not necessarily helpful. This is a UMAP of different types of cells. So again, we said that it may or may not be helpful. These are the stains here. I don't know if you can see it very well. These are the different stages. So you have these at different stages of cell cycle. You have these stains where you have cells in there that are kind of entering different stages of cell cycle. They show this generic graph here where they show, well, this might be helpful. They just kind of show a sort of a cubical cell, a generic cell, but they show the different stages. So you go from precursor to stage 1 to stage 2. So you have precursor, which is this kind of like there's nothing ciliogenetic about it. You have this protein buildup in stage 1, which is S phase, or corresponds to S phase. So you go from G1 to S phase, and you see this little cilia coming out the top. And then stage 2, you get centriole synthesis. So you get a protein buildup in the cell. And then when you kind of enter G2, you get these centrioles that sort of form in the cytoplasm. Then in stage 3, which is sort of more M phase, disengagement, migration, and docking. So you get these little particles in here. Stage 3, and then stage 4, you get ciliogenesis, where it migrates up to the top of the cell and you get these cilia.
Okay, stupid question. Where are these cells from?
This particular type of cell is in mammalian. I think it's in the esophageal tract and the lung. They don't really. I don't know if they mention like a specific organism here.
Let alone a specific organ.
Well, in the respiratory tract, so it's like I guess it's a mix of different organs, but yeah.
Do those cilia move together or they're independent?
I think they move together. I'm not really sure. Yeah. Let's see if they have any other images that might be useful.
I have a vague memory of it having to do with the small intestine.
They probably have there are probably ciliated cells there too. It just basically helps things move in the system. Like in the respiratory system, you have air that's move in and out. In the small intestine, you have, food that needs to move in and out, and so forth.
Yeah.
So, I don't know if they have any really good pictures of the cells. Other than maybe these, where you get like the stains, and you get kind of the morphology of the cells here. Maybe there are a couple.
But you don't clearly see these multicilia.
Yeah, you don't.
Okay, so these are not independent cells like paramecium or something like that.
No, these are just in a cell population. So I guess it's from like a murine model or a mouse model or rat. I don't know. This is mouse. And then they show harvesting from that. Yeah, they work in an organ system. So there are a lot of them in the system. Just looking for some other images. I don't think. Well, this shows in cell cycle, so we already saw this where you have these different stages of morphogenesis in this linked to cell cycle. So as cell cycle proceeds,
these are essentially epithelial cells.
I think they may be epithelial cells. That's why they have sort of the cubical shape, and they're just showing one. They're going to be a population of them.
Yeah, but that's not during embryogenesis. It's during adulthood.
I'm not sure when this happens exactly. Let's see. They mentioned it up here. Yeah, I don't think they mention exactly when it happens. I'd have to dig into the paper more for that. But they may be they may be renewing. Like you may need to have ciliated cells born because they probably die off. And
yeah, this seems more like a repair process.
Yeah, a replacement process. Yeah. So yeah, I don't think there are any other really interesting images here, but just to show that that's a paper I wanted to present to get a sense of that kind of system and see. I know we've been working on this, a lot of stuff with differentiation and talking about what the molecular mechanisms of it are, and so this is a very specialized system, but I think it hits on some of the role of cell cycle and what it's doing in differentiation.
Those cells also have if they're inner epithelium, they'll have also junctions between neighboring cells.
Yeah, they will.
Okay. So is this an individual cell phenomenon or a whole cluster of cells?
Oh, it's going to be a whole cluster. Yeah, there are a lot of cells.
Run through the same steps at the same time.
Well, maybe, but maybe not. I would imagine they're somewhat synchronized. But that's the thing too, is that sometimes cells are, in different cells, you're going to have aberrant processes. Some are going to fail at differentiating properly. Some are going to die during the process. So that's probably, usually they're synchronized, but not often. I mean, one of the things about like working in cell culture is like people synchronize synchronize cells by division. So you don't want cells at different stages of cell division in your culture when you do an experiment. But in tissues, of course, the cells are going to be at different stages of cell division. And I'm not sure about this system. It might be a little bit more coordinated.
Not necessarily. In Drosophila, when a sheet of cells differentiate, they do so in a wave of mitosis.
Right. Yeah, well,
so
that's
coordinated.
Right. And of course, this mentions nothing about the coordination mechanism for. Yeah. All right. Well, that was okay. That was interesting. So, do you have any questions or comments about that? Otherwise, I mean, Dick has asked good questions here. Okay. Any other things you would like to mention before we go? Susan was talking about her adventures in multiphysics.
You might want to have a look at that book, How Life Works.
Yeah.
Okay.
Okay.
It's interesting, but it's sort of self-contradictory.
Yeah. In what way?
It seems to be an attack on the genomic basis of everything based on genomics.
Oh, yeah.
So.
Okay. Yeah.
Oh well. My adventures continue with the COMSOL and other programs. I'm going to take the CompuCell 3D course this summer. It's end of July, early August. And so I'll get a look at that side of things.
Yeah. That sounds good. When does that start?
July 28th, I think, with a Python course, just an overview. And then the 29th that week of the 29th.
Okay.
Anyway, so yeah, I'm hoping to, well, learn something from it, maybe that I can add to my thesis.
Yeah.
I don't know. At least I get a look at the competition.
I put that details on the book in the chat.
Okay, that sounds great. Yeah. All right. Well, I think that's it for today. We'll have another meeting next week. Talk to you then.
Okay. Bye.
Thank you. Bye.
Bye.